Hi everyone, welcome to week 10. This week's lesson is going to be about antimicrobials. There are three types of antimicrobials, or chemical agents that control microbes. And those three are disinfectants, antiseptics, and antibiotics. And we're going to talk about the difference between each of those. So first let's talk about the difference between disinfectants and antiseptics, because those sometimes get confused. So disinfectants are chemicals that are used to kill microbes on inanimate objects. So on surfaces, on tables, these are the products that you would use for cleaning, for example. On the other hand, antiseptics are chemicals that are used to kill microbes on human tissues, so on living skin, for example. Now, some of the chemicals that are used in disinfectants and in antiseptics include phenols, alcohol, and surfactants like soaps and detergents. There's problems with some of them. For example, there's a problem with using alcohol as a disinfectant or an antiseptic. And the problem with that is that alcohol evaporates very quickly. So it has a short duration and acting time on whatever you're using it on, so it can be less effective. For soaps and detergents, there's a difference between soaps and detergents. Soaps physically remove microbes, washing them off. Unless it specifies that it's an antimicrobial soap that kills microbes, soaps are typically just removing the microbes by washing them off. Detergents, on the other hand, kill microbes by destroying their cell membranes. The efficiency of the antimicrobials, or how good of a job they do, is influenced by the following factors. The first is concentration. So a high concentration of the chemical means a better efficiency. But you would need to be careful of the side effects to the person using it. So for example, using 90% bleach is more effective than using 10% bleach on microbes, but you'd have to be careful using 90% bleach because it's more dangerous to the person using it. The second factor is length of exposure. So a longer amount of time that the antimicrobial is up against the microbe, the better the efficiency is. But so if you're cleaning a table, for example, and you spray your antimicrobial and let it sit on the table for 10 or 15 minutes before wiping it down, that would be a better efficiency for that antimicrobial. And remember, that's also the problem with alcohol, is that it has a short exposure because it evaporates so quickly. The third factor is that it depends on the type of microbe. So pathogens that are capable of producing endospores or capsules, for example, are more resistant to antimicrobials because those help them to be more protected. And then the fourth factor is the environment. So an environment with a lot of organic matter makes it harder to kill the microbes because they're more protected by the surroundings. So in a medical setting, things like vomit, pus, blood, all that kind of gross stuff is a good environment for bacteria because all that organic matter is surrounding the bacteria and helping to protect it. So we've been talking about antimicrobials that are used on surfaces. So disinfectants being used on inanimate objects and surfaces, and antiseptics used on the surface of your skin. Now we're going to talk about antibiotics and other chemotherapeutic agents for killing microbes. So the word chemotherapeutic means the treatment of a disease with a chemical substance. So you might see the word chemotherapeutic and think of chemotherapy and think that it applies only to cancer, but really the word chemotherapeutic means the treatment of a disease with a chemical substance. So antibiotics are considered chemotherapeutic agents. So how do antibiotics differ from other antimicrobials? Well, antibiotics and chemotherapeutic agents are used to kill microbes internally, so not on the surface, but inside the body. And there's two categories of chemotherapeutic agents for killing microbes. They're antibiotics. And where do antibiotics come from? Well, they originate from microbes. Bacteria and fungi actually produce antibiotics. And then the second category is synthetic drugs, and those are completely synthesized in a lab. So I just told you that antibiotics are produced by bacteria. Now, why would bacteria want to produce antibiotics that kill bacteria? Well, the answer is to kill off the competition. So producing antibiotics is an adaptation that provides a survival advantage for that bacteria. So they usually kill the ones that are most closely related because they share the most common resources. So there's two categories of antibiotics, broad spectrum and narrow spectrum. Broad spectrum antibiotics kill a wide range of bacteria. For example, they might kill both gram-positive and gram-negative bacteria. And doctors need to be careful when prescribing them because you don't want to take too many broad spectrum antibiotics too often because there is a problem with antibiotic resistance. So narrow spectrum antibiotics are only able to kill a limited group of bacteria. So they might only kill one or two types, more limited. When antibiotics are being developed, there's one major factor for selection as a medical drug, and that's called selective toxicity. And that means that the drug should act against the pathogen and not against the host. So a drug would be selected that kills the pathogen without hurting the person taking the drug. So an example to know, that's a very common one, the bacterial cell wall is often a target for antibiotic treatment. And that's because bacterial cells have cell walls, but human cells don't. So the antibiotic acting on the cell wall is going to kill the bacteria, but it doesn't harm our cells. So we've talked about broad spectrum and narrow spectrum antibiotics and how they only kill certain types of bacteria. We've talked about selective toxicity and how a drug would act on a specific target of the bacteria. So what all this means is that one antibiotic is not going to kill every possible bacteria. And different bacteria are going to have different susceptibility and resistance to the various antibiotics. So in the lab this week, we would do a test to evaluate the antimicrobial activity of an antibiotic, and that's called the Kirby-Bauer test. It's to determine how effective an antibiotic is on a particular pathogen. So to do this test, we would use the Muller-Hinton media, which is a general purpose media that allows all types of bacteria to grow, but it's a loose agar, so it allows for better diffusion of the antibiotics through the media. And it also prevents antibiotics from being broken down. So to do the test, the microbe to be tested would be spread and streaked over the plate, covering the entire plate, streaking a lawn of bacteria over the plate. And then the various antibiotic discs would be placed on the agar. So there are these paper discs that each contain an antibiotic. And then after growing in the incubator, you would measure the diameter of the zone of inhibition around each disc. So that would be the area where the bacteria can't grow due to that antibiotic. So you'll see that some antibiotics give you a big zone of inhibition, some give a small zone, and some give no zone at all. So if the bacteria is resistant to that antibiotic, there would be no zone of inhibition. An intermediate would be a small zone of inhibition, and if it's susceptible to the antibiotic, you would see a big zone of inhibition. You would then measure the zone of inhibition for each of those antibiotic discs on your plate, and then look at this antibiotic sensitivity chart for your microbe to determine if it's resistant to each of those antibiotics, if it's intermediate, or if it's susceptible to the antibiotic. And to give you a little bit more information about the effectiveness of the antibiotic and the concentration at which it's effective, you can do another test called the minimum inhibitory concentration test. And that uses a strip with different concentrations of the antibiotic. As you can see in the picture, the different concentrations of the antibiotic are marked on each line of the strip. So what does that tell you? It tells you the exact range of concentration that's needed to kill that bacteria. So if you look at the picture, you can see the minimum concentration at which the antibiotic starts to be effective, and you can see the most effective concentration range for that antibiotic on that microbe.